for today, we're going to be beginning our discussion on microbial metabolism. Metabolism is defined as the sum of all of the chemical reactions that occur within a cell. It's comprised of two major branches, catabolism and anabolism, where catabolism is breaking down large molecules into smaller ones to release energy, and anabolism is the synthesis of complex organic molecules from simpler ones, requiring energy. So you can think of catabolism as those hydrolysis reactions where we are breaking down larger molecules, breaking bonds, and therefore releasing energy from those molecules that we've stored energy in, where anabolism comes in line with those dehydration synthesis reactions, which do the opposite. We join up smaller molecules and form bonds, and within those bonds that we're forming, we store energy. Microorganisms can work from a variety of different nutritional sources. These can be different kinds of sugars, they can be lipids, they can be proteins. It doesn't matter, but compared to eukaryotic cells, they can work from things that are not typical for larger, more complex organisms, of course. Most of the metabolic pathways you'll see will converge on the production of pyruvate. From there are many different options depending on what types of environments bacteria live in or what their preferred metabolic pathways might look like. They can undergo fermentation, where they're going to produce byproducts like lactic acid or alcohol. These typically occur in anaerobic environments where oxygen is not present. Or you can have pyruvate being shuttled off to something like the Krebs cycle, where it will undergo a lot of different reactions to produce ATP, but also other cofactors that end up in our electron transport chain. The Krebs cycle is your best bang for your buck per pyruvate molecule, but some bacteria do prefer this fermentation method instead. I just also wanted to point out that it's not only pyruvate that can flood into the Krebs cycle and cause that to go. You can have a variety of different amino acids or lipid molecules that can act as precursors at different steps along the way through that large cycle. So while it's ideal to start with pyruvate because you get the most out of it with all of the different changes that happen to that molecule throughout, you can start partway through with other cofactors instead. But at the end of the day, the biggest goal for any metabolic pathway is the production of energy, and usually that molecule is going to be ATP or adenosine triphosphate. Why do we need energy? Well, it's the capacity to do work. In order to do anything, whether it's division, DNA replication, transport work, migration, mechanical work, enzyme work, anything that a cell is undergoing requires some form of energy. So all of these metabolic pathways and conversions of molecules into different types of intermediates, and then the byproducts that arise from those, whether it's carbon dioxide or whether it's oxygen or whether it's energy or NADH or FADH2, it doesn't matter. All of those molecules contribute to the production of ATP, which will allow cells to be able to function. This diagram summarizes it up pretty well, the three different kinds of work. Mechanical work is going to be cellular movement. So you can see an example here of a cytoskeleton and some motor proteins here that are moving components throughout the cell. You can also see down here transport work, where we have transporters and other proteins at the cell surface that need energy. We talked about this with active transport to move stuff across the membrane. This happens very often. There is also chemical work where we're synthesizing things or we need to facilitate a reaction of some kind. And the phosphorylation of different molecules will allow for these reactions to proceed in a lot of cases. So whether we're talking about mechanical, transport, or chemical work, all of those things that fall under those categories allow cells to do the work that they need to do and to exist and continue to exist. We always talk a lot about storing energy in bonds, and adenosine triphosphate is probably the example that we talk about the most. When it is in its full form as ATP, you can see that there are 3 phosphate groups that are attached at the side of this molecule here. These bonds that are formed between these phosphate groups contain energy, and when they are broken, energy gets released. So typically when you see the conversion of ATP to ADP, what's happening is you're cutting off or breaking a bond between this second and third group, and that bond releases energy, which can then be used for all of the things that we've been talking about. So this example shows that exact reaction. You can see that we've got our 3 phosphates. This is ATP. And depending on what reaction you're doing or what kind of work is being done, you're going to see the breakage of that bond to form inorganic phosphate and adenosine diphosphate, or ADP. And again, that good byproduct that we want to see arise from these reactions is that energy. Now, shifting gears here, when we talk about metabolic pathways, what we're really talking about are enzymes. They are what make metabolism happen because they're responsible for the conversion steps of different types of substrates throughout the whole reaction. They're going to produce a bunch of different intermediates along the way that can then feed into the next reaction, and so on and so forth. Enzymes can be quite simple or complex depending on what they are, of course, but what they all have is an active site that binds a substrate. So when we look at metabolic pathways, the substrate is going to be one of the intermediates in the pathway that's going to bind onto that enzyme and become catalyzed or changed in some way. So this example down here shows an enzyme with an active site that's meant for this disaccharide here of fructose and glucose, which is sucrose, as you might know. And what it does is comes in here, and you'll see that the substrate binding sites, or the active site on the enzyme, is specific for the substrate. And then when the substrate binds, there is a reaction that's catalyzed to split them. And you'll see here the splitting is done. And at the end of that, you end up with glucose and fructose, which has been separated from one another. The enzyme can then bind a new sucrose molecule and continue to do that work. So enzymes are not consumed or changed at all in these reactions, but rather act as a machine that you can keep feeding things into over a long period of time to get the same kinds of products produced. There are also the ideas of temperature and pH very heavily affecting the efficacy of enzyme reactions. For both of these parameters, there is an optimum that enzymes will work best at. And if you go outside of that range, either above or below, what you'll find is that the enzyme activity rates will drop off. You can see that down here in these two diagrams where we've got pH optimum at the top here, temperature optimum at the top here. And as we move away from those in either direction, you'll see that those rates are going to drop away. For pH, what we see is a change in the biochemistry of amino acids that are involved in the active site of an enzyme. So they can change and become less compatible with their substrates, or you'll have a physical conformational change in that active site, which will prevent that enzyme from being able to bind its substrate as well or at all. Temperature, on the other hand, can cause denaturation or unfolding of components of that active site. And again, those physical differences can cause problems with enzymes binding to their substrates and catalyzing the reactions that they're meant to do. When we look at regulating enzyme activity, there's a number of different ways that we can do this. It can be at the level of transcription or at the level of translation. And what that means is if we're working at the level of translation, the protein's already been translated. It already exists in the cell. And at that point, you can control what that enzyme is doing in a variety of different ways. So if this over here is our enzyme reaction, and you can see a substrate and a product, if we're controlling it post-translationally, it means that we're affecting the enzyme's activity. So changing the temperature, changing the pH, maybe we're changing the amount of substrate that's available. Maybe we're changing the amount of enzyme that's available. All of these are going to reduce the amount of product that is being made from these reactions. There's also the ability to regulate how much of this protein gets translated at all. If we prevent the process of the mRNA being translated into protein, then we're not going to have any enzyme synthesis because the enzymes simply don't exist. Or you could go even further back to transcriptional control, where you're not even going to produce mRNA, which would code for that enzyme to begin with. So you can manipulate metabolic pathways, either in the laboratory, if you're trying to study a metabolic pathway, but your cells will often do one or many of these things to regulate their own metabolism and to kind of streamline it based on what is needed at the time. Because don't forget, if you have a situation where you've got energy being expended for metabolism or being produced by metabolism, what you don't necessarily want to have are these reactions just running for the sake of running. There's a high level of control to determine how often they run, how much they run, and these are some very generic examples of what can be done to help to curb that process from just running for nothing. One of the regulatory mechanisms is called feedback inhibition, and this is when we have a lot of product that's being produced downstream, so further down in a long chain of reactions. Let's say in glycolysis, it's like step 8 or 9. And the production of a lot of that molecule will actually go back and inhibit earlier steps in the metabolic pathway. And you can see an example of that here, where we've got an end product coming back and preventing the activity of one of the earlier enzymes in this reaction. This is a mechanism by which we can limit how much of this reaction will actually occur. Because again, we don't want to just keep accumulating stuff that we don't need since these processes are very involved. So having a means to shut that system down is very important. The way that this happens is through a process called allosteric regulation, where you've got not the active site being interfered with on an enzyme, but a secondary site. So if you have a look here, we have our active site where our substrate would normally bind, but the allosteric site is a secondary site. We have a molecule that can come on and bind this area, which will change the active site. You can see in this example, when inhibition has occurred, this active site is changed. There's a physical change in that conformation that will prevent that enzyme from binding its substrate and doing its work. When there is no binding, of course, that activity proceeds normally. So molecules from further down in that reaction will come back up earlier to enzymes that are working ahead and will bind to them to prevent them from being able to do their job. Now, in disease states, sometimes these regulatory mechanisms aren't working very well, so we can employ chemotherapeutic interventions that can bind to enzymes and inhibit their activity. We can do something called competitive inhibition, where we've got something competing directly with the substrate. So you can see here, competitive inhibition involves another molecule that's going to come in and bind to this substrate binding site on the surface of this enzyme, preventing the normal substrate from being able to bind. The normal substrate will bind sometimes, but sometimes it won't because this other molecule will be in direct competition with it. Or, like we just discussed, you can have that non-competitive inhibition, allosteric inhibition, where you have a molecule that is introduced that will bind to a secondary site to again inhibit that reaction. Substrates can no longer bind to their normal active site, and that enzyme activity is lost. We're going to look here at some catabolic reactions that are again going to involve the breakdown of larger molecules to produce energy in order for cells to do their work. These are again those hydrolysis reactions where we are breaking bonds and releasing energy. There are 3 different kinds of reactions here to consider. There's chemoorganotrophy, chemolithotrophy, and phototrophy. And in all of these examples, what we're doing is harvesting energy from different sources and converting that into chemical energy, which can then be used to do work at the end of the day. And we're going to look at some examples of these coming up. So no matter which one of the reactions that we're looking at today, there are 2 ways to produce energy. We can do what's called substrate level phosphorylation, and this is done in the absence of oxygen. So this is, for example, done in anaerobic respiration or fermentation reactions, where we have ATP being produced as a byproduct of the normal steps of a metabolic pathway. So if you take a look up here, you can see that the pathway is proceeding as normal. We have substrates and intermediates being changed along the way. And then as a byproduct of one of the reactions, we get ATP that gets produced. So substrate level phosphorylation means that it is a native way that ATP is getting made just from the metabolic pathway running on its own. The second is oxidative phosphorylation, and this is where we're having the production of ATP in aerobic conditions, so in the presence of oxygen at the expense of the proton motive force. This is done specifically through the electron transport chain. We'll start first with fermentation because it does not use the electron transport chain and does not require the proton motive force. This is again because we're having these reactions occur in the absence of oxygen. And if you recall in the electron transport chain, as the electrons are being passed along from complex to complex, at the end of that is oxygen as the final electron receptor. So when oxygen is not present, the proton motive force is not going to be used in these cases, and we're only going to be relying on substrate level phosphorylation. So you can see in these examples, we have lactic acid fermentation and alcohol fermentation. We have glucose running down to pyruvate. So all of these little dashes in this arrow, in both cases, are referring to the steps of glycolysis. And you'll see along the way, all that we're really producing are 2 ATP. So when we consider how much more ATP can be made if pyruvate goes off to the TCA cycle or the Krebs cycle and goes through all of those processes, to have 2 ATP come out of this reaction alone is not very much. So fermentation does still yield energy, but not nearly as much or as effectively as it does in aerobic conditions. Microorganisms will produce a lot of different byproducts through fermentation. You can have alcoholic or lactic acid, like we just discussed, but there are also formic acid fermenters that will end up producing mixed acids or butanediol fermentation as well. So, depending on what you're starting out with, what types of enzymes are present, what types of environments these microorganisms are going to live in, fermentation will differ depending on what the needs are or what the environment will allow them to be able to do. On the level of industry, we can take advantage of a lot of these different types of fermentation for our own benefits. So you can see, and these are just some examples, of course, I'll never ask you to identify what types of byproducts Clostridium species produces, for example, but you can see that after we end up with pyruvate, that can be fermented into a variety of different things by a variety of different organisms, producing lots of different things that we use on the daily. Things like carbon dioxide or propionic acid, lactic acid, ethanol, acetone or isopropanol, or acetic acid. So all of these different byproducts obviously benefit us, but they also benefit the bacteria that are making them in their environments as well. We just happen to take a look at that and say, that looks tasty. Let's make that happen in a factory so that we can eat these delicious foods. You probably heard it a bunch along your lives about not eating sugar or else you'll get cavities. And this is true to an extent, but it's not actually the sugar itself that causes tooth decay. It is actually the microorganisms that live in our oral cavities undergoing fermentation reactions and producing acids from those sugars that cause our teeth to become damaged over time. As these acids get deposited and deposited, they tend to wear away at our tooth mineral until that hole eventually reaches the nerves underneath, which can cause pain and obviously problems, which would result in needing some kind of dental filling, or if it's really severe, having a tooth removed altogether. So brushing your teeth and making sure that you're flossing regularly is important to not only prevent cavities, but to prevent extreme cases of periodontal disease, where you can end up with inflammation and other kinds of bone loss problems over time from damage that will eventually not be reversible. There are also several different kinds of respiration pathways that occur in bacteria, which are completely dependent on what kinds of nutrient source that a particular species will enjoy. We've already talked a bit about aerobic respiration, which is going to use oxygen as the final electron acceptor, but there are also anaerobic respiration conditions where other molecules are also suitable to receive electrons at the end of the electron transport chain. So it can happen in the absence of oxygen. It's just most efficient in the presence of oxygen. So you can see in these examples, we have organic energy and an electron source, and everything's the same. It goes through the same processes in both cases. We get to the electron transport chain, and the only thing that differs in these two examples is that electron acceptor at the very end in aerobic respiration will be oxygen. In anaerobic respiration, it can be a variety of other molecules. When we look at aerobic respiration, this process is going to occur in 3 stages. We have polymers. These can be ways that glucose or other types of sugars were stored away. They can be proteins, they can be lipids. It doesn't matter what the starting molecule is, but they're polymeric and they get broken down into monomers via hydrolysis reactions, right? We're breaking bonds, we're making smaller molecules from larger ones. And then the monomers are broken down into even smaller molecules, which produce products like minimal amounts of ATP, NADH, and possibly FADH2, which eventually culminate in the production of pyruvate. Pyruvate then gets shuffled off into the tricarboxylic acid cycle, or the Krebs cycle, where a lot more ATP is produced, as well as even more NADH or FADH2. Those can then go off to the electron transport chain. So you can see from this diagram that no matter what your starting material is here, you can start from larger molecules that get broken down into smaller ones. Those intermediates are going to get fed into various parts of the glycolytic cycle, sometimes even in the TCA cycle. And over time, you're going to have the production of other cofactors that can then go on to be electron donators to the electron transport chain to produce even more ATP. You'll see along the way too, a little bit of ATP is going to get produced natively, but the big bang for the buck comes from the production of these other cofactors that will donate their electrons to the electron transport chain. Now, depending on the species, glucose metabolism can happen in a couple of different ways. These are all considered to be glycolysis because they're starting with glucose and terminating in pyruvate, but the intermediates along the way are going to be different. So we have the Embden-Meyerhof pathway, the pentose phosphate pathway, and the Entner-Duoderoff pathway. And we'll talk a little bit about each of these. When we look at the tricarboxylic acid cycle, this is typically the third stage of aerobic respiration. And this entire process's purpose is to oxidize pyruvate to produce carbon dioxide as well as NADH and FADH2. So from every molecule of glucose that gets catalyzed in glycolysis, we have two pyruvates that are formed. So for every one molecule of glucose, we're having two runs of the TCA cycle. So you can see here at the top, pyruvate is again the end product of glycolysis, any of the three pathways that we already discussed here. There's a conversion into acetyl-CoA, and then that's going to run through the entire cycle, and all of the enzymes along the way are going to change these different intermediates into more and more. And the important steps here, you'll see, are highlighted. We have the production of NADH in several steps here, FADH2, over here, and a final NADH out towards the top. So it's these molecules that are then going to go on to the electron transport chain to donate their electrons to create ATP from that proton motive force that gets generated from the electron transport chain. Now, you might recall that the TCA cycle occurs in the mitochondria in eukaryotic cells, but because prokaryotes don't have a mitochondria, their TCA cycle occurs in the cytoplasm. Depending on what the requirements are for a certain bacterial species, they might not even have a complete set of TCA cycle enzymes, because if they're not going to be in aerobic conditions very often, they're not going to need to undergo this process too much. They may not need to produce those enzymes or keep them available, or even have genes that code for them. Most species do tend to have some, though, because there are intermediates from the TCA cycle that are really important in the biosynthesis of other types of molecules like proteins or lipids, for example. So the NADH and FADH2 that get produced in glycolysis and the TCA cycle are going to act as electron donors in the electron transport chain. So the electron transport chain undergoes a series of redox reactions where electrons are being transferred from complex to complex, and this as a result will establish the proton motive force. So as complexes are passing along these electrons, they're also pumping protons across the membrane to create a proton gradient. There are 4 different complexes in the electron transport chain. There's complex 1, 2, 3, and 4. You'll notice here that complex 2 is the only one that does not pump protons across the membrane. 1, 3, and 4 do, but 2 does not. So if we take a look here, the different complexes have been lined up based on their electronegativity, meaning that the more electronegative molecules are going to be attracting those electrons to come towards them. So you can see here, we've got complex 1 and 2, 3, 4, and at the very end, of course, in aerobic respiration, we have oxygen as that final electron receptor. So you can see that NADH and FADH2 are going to give their electrons into this process, and they're going to travel along these different complexes, and over time, they're going to be pumping protons across the cell membrane to create that gradient that can then be used by ATP synthase, the large protein that produces ATP, in order to form more of that molecule. When we look at where the electron transport chain is located in prokaryotes and eukaryotes, it will vary. In eukaryotic cells, the electron transport chain proteins are found within the inner mitochondrial membrane, and the proton gradient gets established in the intermembrane space. So if you recall, mitochondria are double-membraned organelles, and there is a little place in between those two membranes called the intermembrane space. So if the proteins are embedded in that inner membrane, and they're pumping protons across from the mitochondrial matrix into the intermembrane space, then that proton gradient is going to form between those two layers. In prokaryotic cells, the electron transport chain is found in the plasma membrane, and that proton gradient gets established between the periplasmic space and the cytoplasm, no matter what the gram status, right? They all have that inner membrane or a single membrane, depending, and they're going to house the electron transport chain complexes. So here's another diagram showing eukaryotic cells and their electron transport chain. So this is the mitochondrial matrix on the inside, the intermembrane space here. There's a second membrane over here if you want to picture that. We have complexes 1, 2, 3, and 4. You can see as well that as different intermediates are donating their electrons, that we're having protons pumped across. You'll also notice an easy way to remember which complex does not pump protons is that complex 2 here is not a transmembrane protein. It's only affiliated as a peripheral protein with the inner leaflet of that membrane, so it's not going to be able to pass stuff through. You can see here at the end as well that we have ATP synthase, where we're going to have the production of ATP from harnessing some of those protons that have been pumped across and using them to produce ATP, but we'll talk about that in a bit more detail shortly. When we look at ATP synthase, which as its name implies, synthesizes ATP, it's made of two components. We have the F1 and the F0 parts. The F0 component is the one that's embedded into the membrane, and it's going to act as a proton channel. And then down below we have the F1 portion, which is almost like a motor protein. It's attached by a stalk, this little purple guy here, but you'll find that the F1 portion is going to be internal, so none of it is membrane bound. In eukaryotic cells, it'll be found within the matrix. In prokaryotic cells, it will be found within the cell's cytoplasm. 3 protons are required to drive ATP synthase, and it works like a rotary engine. You have the F1 and the stalk, which will move, and that conformational change in the F0 protein will cause for binding of ADP, an inorganic phosphate at that catalytic site, synthesis of ATP, and then its release after the fact. In organisms that do need to use that proton motive force for other processes, that proton gradient is actually reversible. ATP synthase can move protons backwards through the membrane when it's required. So if there's a proton motive force required for something like motility, ATP synthase can be used to recreate that or reestablish that in order for other processes to take advantage of it. The maximum ATP yield that we get from all of these processes in aerobic respiration is about 36 to 38 molecules. Here you can see in glycolysis, we have a couple of different ATP that get produced, which would then be the total that would get produced in something like fermentation. But if you send your pyruvate molecules into the TCA cycle and produce all of these different cofactors that go through oxidative phosphorylation with the electron transport chain, you do have a significantly higher amount of energy that gets produced in these conditions. Now, I just have a couple of examples of how different macromolecules can get broken down and feed into these processes to begin with. When we're talking about polysaccharides, we're going to see them getting cleaved by exoenzymes. These can be secreted by prokaryotes or fungi, and they will actually degrade or break down these large sugar molecules into smaller ones that can be taken up by cells and then fed into these metabolic pathways. Many microorganisms will actually store polysaccharides until they need them, and then when they're required for energy, they'll bring them out and start to break them down. Some examples of enzymes that do this work are amylase and cellulase that break down glycogen and cellulose, respectively. And these can both be used to produce different types of sugars that can feed into these other metabolic pathways that we've been discussing. Lipids can also get broken down into smaller molecules, and this can be done by lipases, which are enzymes that break down triglycerides into their component structures like fatty acid chains and glycerols. From there, they can get oxidized further into smaller intermediates that can enter different stages of cellular respiration, like the tricarboxylic acid cycle or other metabolic pathways where they would be one of the many intermediates along the way. Proteins and amino acids are also a very big source of sugars because the only difference in some of the structures between amino acids and sugars are the presence of amine groups. So if you are going to need an intermediate for one of your sugar pathways, you can just cleave off the nitrogen and end up with an intermediate that can enter one of these sugar pathways instead. So one of the first steps is to remove that amine group through a process called deamination. Deamination can be done through a process called transamination, where you pull the amine group off of one molecule and stick it onto something else. So let's look here at an example. We have alanine and we have alpha ketoglutarate. If we pull the amine group off of alanine and stick it onto alpha ketoglutarate, we end up with pyruvate and glutamate. So just with a couple of little changes, some subtle little changes in the molecular structure of some of these very common intermediates, we can generate pyruvate and get a bunch of energy out of that. So, the fluidity of metabolism is really interesting and really endless, and it's so adaptive that no matter what, most microorganisms, so long as they have a nutrient source, can make do with it based on these little changes that can be done.